Hi, I'm Jessica O'Neill, and this is my final presentation for my genetics lab, BY212, for the spring 2023 semester. This semester, we worked with the microbiomes of submerged plants. My specific project was on the effect of poor quality growing environments on different submerged plant species microbiomes. So an important background when looking at this research is knowing the different parts of the plant microbiome. The phyllosphere includes the outer portion of the stem and leaf of the plant. The endophytic microbiome, which is what I was looking at and our lab was looking at, is the inside of the stem, leaf, fruit, seeds, just any part of the plant. The different cells, viruses, bacteria, anything that's found within it. And the final part of the microbiome is the rhizosphere, which includes the outer portion of the root area of the plant. The microbiome holds important endophytes to plants. These endophytes can be either helpful or harmful to the plant, depending on whether they're pathogenic or not. The enzyme our lab was looking for in particular is ACC deaminase. This is helpful in the promotion of plant growth, and our specific lab was looking just to find samples of it, but eventually to find ways to apply it to different crops to help promote growth in them. The three samples that I used were labeled SO1, SO2, and SO3. My hypothesis was that SO3 would have the largest amount of diverse endophytes within its microbiome because it had the harshest environmental conditions to thrive in. As you can see in the pictures below, SO1 and SO2 both had clear water along with plants growing within them with ease. SO3, on the other hand, had evidence of gasoline within its water, along with an abundance of dead plants around it, so to me suggesting that it had to survive harsher conditions. Our methodology throughout the semester was that we first collected our plant samples. I collected samples of the entire plant, including the stem, leaf, and root sections, along with water samples from the area to have the plant stay in while I was working on processing it. To process the sample, we first used soap and water to wash the plant 3 times over. Then we made 70% ethanol and 2% bleach to wash the plant in. This was to get the, to just kill off any microbes in the phyllosphere of the plant, since we were only interested in those endophytes within the plant. We then rinsed the plants again with water just to make sure we had gotten off any evidence of that ethanol and bleach so we weren't accidentally killing the endophytes. We then placed it in the phosphate buffer system, the PBS buffer, along with crushing those samples so to make a solution to then culture. Our next step was culturing samples of each of our plants. We did this by spreading the solution we had created during processing over both 100% and 1% LB media plates. The 100% and 1% was selected because of the varying nutrients each plate would have. It would be much harder for the bacteria to grow on 1% than it would, or 1% plates than it would be on 100% plates because they have far less nutrients. Once we had cultured the bacteria on the 100% and 1% plates, we then selected samples from each plate to do a pure culture of. Once these pure cultures had been allowed to culture for a couple of days, we did microbial amplification in order to do PCR or polymerase chain reaction. We needed to do this in order to amplify the DNA that was present from the different samples. When we amplified the DNA through PCR, we were looking for the 16S ribosomal DNA. This was important because we could then run that data once, making sure we had cut out any redundancies through the basic local alignment system tool or search tool, right? Or BLAST, which can be used to find the bacteria or microbe enzyme that our DNA sequences were associated with. This brings me to the results of BLAST. It confirmed my hypothesis that SO3 did have the most different endophytes within it. 6 out of 7 of my pure cultures from SO3 were endophytes. One sequence was so short that BLAST was unable to find a significant similarity found within it. In addition, with my results, 8 of 15 samples that I collected were, or samples that I cultured were pathogenic to humans. These were pathogenic in varying degrees. The least ones were pathogenic to humans who were completely immunocompromised and had no way to fight back against them, and some were pathogenic to humans, no matter how strong their immune system was. All 15 of my samples had beneficial properties to plants. The most common property the endophyte had was promotion of plant growth. However, they all did this in different ways. In addition, 10 out of 15 of the samples, or 2/3 of the samples that I collected or cultured, had records of ACC deaminase being present within their species. This is why the most common property was promotion of plant growth, because ACC deaminase, which was found in most of the species that I found via BLAST, promotes plant growth itself. So it makes sense that these promote plant growth. And then I had one sample, 014, that was too short to have any significant similarity found within it between other sequences in BLAST. And I had one sample, 012, that was lost during the process of PCR. And then here are my citations. Thank you.